Well, this is our final segment of material on the gastrointestinal system. And in this one, we'll be focusing on regulation of gastrointestinal functions. The first part will be emphasizing general aspects of regulation, and then in the second part of this lecture, we'll be focusing on specific details. So first of all, the regulation of GI function is not based on the concept of homeostasis, because the system is designed to maximize absorption regardless of whether the nutrients are needed. This is unfortunate. It's one reason we gain weight so easily. But it also stops the system from being less than maximally efficient when it comes to absorption of nutrients. And another aspect of regulation, of course, is to regulate the conditions in the lumen of the GI tract to maximize absorption. So it's not only not based on the concept of homeostasis, but it is not based on the concept of balance, for that matter, because it is biased toward maximizing input regardless of output. There are neural and endocrine pathways that are involved. The enteric nervous system is a layer, two layers actually, of mesh-like layers of neurons, and it's hard to see in this figure. The main thing here is just to give you a general idea of that it's in the wall of the GI tract. There's a submucosal layer, which of course is, as the name suggests, right underneath the mucosa, and a myenteric plexus, which is more exterior underneath a layer of connective tissue and smooth muscle. Both of these plexi, in plural, plexus and singular, work to mediate reflexes through what's called the enteric nervous system and control many GI functions. We don't actually need extrinsic neural control, that is nervous control from the autonomic nervous system, to allow digestion and absorption to go on. However, the autonomic nervous system can modify the responses of the enteric nervous system. So the CNS does contribute to the total neural control, generally because of the autonomic nervous system inputs coming in. There are hormones in the GI tract that are secreted from endocrine cells in the stomach and small intestine. We've talked about some of these in terms of acting as enzymes. In fact, some serve a dual role as both an enzyme and hormone, and some of them just function as hormones, as well as there are neural sensory receptors in the GI tract that detect the environment and initiate reflexes. Some of these reflexes, of course, through the enteric nervous system, and others through the central nervous system. So let's take a look at the hormones. This table 20.2 in your textbook lists the four major hormones, that is gastrin, cholecystokinin, abbreviated CCK, secretin, and glucose-dependent insulinotropic peptide. We'll talk about each of these individually. Gastrin is secreted in the stomach, and the stimuli for secretion are proteins and protein digestion products in the stomach. So these gastrin release is initiated in part by chemical stimuli, as well as physical distension of the stomach and parasympathetic input to the stomach. The actions of gastrin are to stimulate gastric acid secretion and motility, to stimulate ileal motility and reflexes of the ileocecal sphincter, and to stimulate mass movement of the colon. So when the stomach receives food, and we start to have initial breakdown of some of these food products, as well as even parasympathetic input coming from the brain when we see or smell food. These are all going to initiate digestive processes. So gastrin is a substance that actually enhances the digestive process. Cholecystokinin is released by the duodenum and the jejunum, and it's stimulated by protein or fat digestion products in the duodenum. And its function is to inhibit gastric secretion, gastric secretion and motility, potentiate the actions of secretin on pancreatic bicarbonate. Secretin is what we'll talk about down here. stimulate pancreatic enzyme secretion, stimulate bile secretion by the liver, and to stimulate gallbladder contraction and relaxation of the sphincter of Oddi. So cholecystokinin inhibits gastric function but stimulates function of the lower portion of the GI tract. And it's a way of coordinating the activity of the different segments of the GI tract. If you think about it, gastrin is starting the whole process. Cholecystokinin is giving some feedback telling the entire system that the stomach need not continue to send food downstream because the small intestine is now processing it, and it would be premature to send more food down from the stomach to receive more intestinal processing. Secretin is released at the duodenum and jejunum, and it is released when acid is entering the duodenum. This, of course, is a signal that chyme is coming into the duodenum from the stomach because of the presence of the acid. And because of that, it makes sense that it would then send feedback to the stomach to inhibit gastric secretion and motility, to stimulate pancreatic bicarbonate secretion, to potentiate the action of CCK, which we've already said gives some negative feedback to the stomach, on pancreatic enzyme secretion, and stimulate bile secretion by the liver. So just as the food moves sequentially through different segments of the GI tract, we have hormones that are being released sequentially that are giving both feed forward for downstream to prepare lower segments of the GI tract for receiving the incoming material, and negative feedback to the preceding segments of the GI tract to slow down the processing so that the chyme is not ushered further down the GI tract before the lower segments are ready. And then glucose-dependent insulinotropic peptide. Now think about the name first of all. It's dependent upon glucose. Insulinotropic. It's acting like a tropic hormone. So it's going to stimulate insulin release ultimately. It's released in the duodenum and the jejunum, and it is released as one might expect if it's going to have an insulinotropic effect by glucose as well as fats or acid in the duodenum. So of course, again, it's signaling that food has entered the duodenum, or chyme has entered the duodenum coming from the stomach, as well as distension of the duodenum. So it's both chemical and mechanical stimulation of the duodenum. And again, it's going to give negative feedback. It's going to inhibit gastric secretion and motility and stimulate insulin secretion by the pancreas. So, excuse me, there's a lot to memorize, obviously, in terms of the stimuli and the actions. But if you think about what they're doing, each of these hormones, as I said, they're being released in a sequential manner as food material is moving further down the GI tract. And with that, they're going to be stimulating their own segment and the segments further downstream into activity to prepare them for receiving the food material, but sending negative feedback to the previous segments to help slow the processing of food material so it doesn't keep pushing more of the chyme downstream before the lower parts of the tract are ready to receive it. Excuse me. Now with this, there are both short-term and long-term aspects to the regulation of food intake. Short-term are what we call satiety factors. That is, how satisfied are you? Have you filled yourself up, or are you still hungry? The factors that control this are insulin, cholecystokinin, as well as neural input from both mechanically sensitive and chemically sensitive receptors in the stomach. So these are basically, the short-term factors are controlling your eating during an actual meal and whether or not you've become full, or at least what we commonly describe as becoming full. Long-term, in terms of how much are you eating overall from day to day. And there the substances that are involved is leptin, which is a hormone that's released from adipose cells, that is fat cells, when calories exceed demand. So if a person is consuming more calories than they need, they're of course adding the amount of fat that is deposited in these adipose cells, and this causes the release of leptin. And leptin is supposed to suppress hunger and increase metabolism. Now I say it's supposed to because obviously if it worked beautifully, none of us would ever gain weight. But it does have its limits. There are some, at least animal models, where leptin is not properly released, and those animals, in fact, do become tremendously obese. But leptin is not something that is a tremendously powerful signal for satiety. And that's one reason that many of us, in fact, still do overeat. And as I said, there's both neural and endocrine control. So we have various stimuli that are going to be stimulating chemoreceptors, mechanical receptors, osmoreceptors, and they're going to do one of two things. They're going to activate what we call a short reflex pathway, which basically is going to work right there on the enteric nervous system. So you've got a stimuli coming from the lumen, activating receptors that are right there, actually part of the enteric nervous system, and it's going to cause activation of the effector cells within the enteric nervous system, which will do a couple of things. Actually work on smooth muscle or glands, causing some sort of a response, as well as stimulating nearby endocrine cells, which are going to release hormones into the blood. And some of those hormones will, in fact, affect some of these effector cells themselves. Long reflex pathway is called long because it's going all the way up to the central nervous system. And other things can interact with it, emotional states. As you may know, if you're very upset, your appetite is suppressed. Actually, eating might make you feel nauseous because of the stimulus activating with some of these receptors, interacting with these emotional stimuli, could give you the sense of indigestion or nausea. Other things, the sight, smell, or taste of food. Suppose you really like pizza and you just sit here looking at some pizza that's steaming and smelling ever so good. What's going to stimulate the central nervous system, which is going to, through the autonomic nervous system, actually activate digestive processes even before you've started to consume the pizza. Of course, it's preparing your GI tract for the entry of the food. So long loop reflex pathway involves, in terms of GI function, involves the central nervous system. Short reflex pathways involve the enteric nervous system. And of course, they both do interact at the level of the enteric nervous system, because the long reflex pathway ends up coming down through the autonomic nervous system to alter the activity of the enteric nervous system. Now, with these last two slides that I've shown, I'm beginning to get into the idea of phases of gastrointestinal control. And there are three phases: cephalic phase, gastric phase, and intestinal phase. And as I said a moment ago in the previous slide, just stimuli that originate in the head, and what we mean by that is just even thinking about food, tasting, smelling, seeing it, is going to actually, is what we call the cephalic phase, it's going to actually help to start some of the digestive processes. It's going to increase secretion and motility. You'll start salivating. Your stomach will start becoming more active. And it's, of course, this is in preparation for consuming food. The gastric phase, the stimuli originate in the stomach. And these involve the short and long loop reflexes that we talked about before, as well as GI hormones. Now, the gastric phase is going to do two things. One is the stomach is going to be signaling downstream to the intestine that material is on its way, as well as so that's going to be feed forward. It's going to give feedback. It could actually, as the stomach becomes more distended, as you get fuller, it can actually inhibit input coming from the cephalic phase to reduce salivation, to reduce the desire for food. Then the intestinal phase, as you might have already guessed, means the stimuli originate in the small intestine. And again, this involves short and long reflexes as well as hormones. And as I said, for the gastric phase, in this case, there's nothing to give feed forward to, but it's going to give feedback. It's going to help inhibit gastric activity as a way of decreasing the amount of food coming down to the intestine to give the intestine more time to process the food material that it has. Just as the feedback from the stomach to cranial structures is going to help reduce activity there to give the stomach more time to process the food that it has. So each of these phases interacts with the other phases. They have important functions within themselves, but a tremendously important part of their function is to integrate with the other phases. So now let's take a look at some specifics of regulation of GI function. We'll talk about secretions first. Secretion of saliva. The taste and the texture of food stimulates both mechanoreceptors as well as taste receptors or taste buds. We didn't talk about this in the neural section, but most of you have heard of taste buds. Taste buds are organs that contain taste receptors. These, of course, react to, again, the chemistry or the taste of the food as well as the texture, stimulating these receptors. And these send input to the salivary center in the medulla oblongata in the brainstem that is going to activate the autonomic nervous system and stimulate salivation. Again, it makes sense. The presence of food in the mouth stimulating salivation because that is going to initiate the digestive process. The autonomic input to the salivary glands is both sympathetic and parasympathetic. The parasympathetic system produces a very watery saliva, whereas the sympathetic nervous system produces a more mucus type of saliva that is much thicker. For gastric acid secretion, there are parietal cells that produce hydrochloric acid. We've mentioned this before. The enzyme carbonic anhydrase catalyzes the production of bicarbonate and hydrogen ion. The hydrogen ion is then actively secreted into the lumen of the stomach, and bicarbonate, as we mentioned before, is transported into the interstitial fluid in exchange for chloride. The chloride diffuses into the lumen of the stomach through the channels, and of course the chloride combines with the hydrogen ion to produce hydrochloric acid. The net result is acid production. This hydrogen ion and chloride are secreted into the lumen of the stomach, and bicarbonate enters the interstitial fluid and then the blood. So we have an exchange of bicarbonate going into the bloodstream in exchange for acid that is being produced in the lumen. The acid secretion of the stomach is regulated by the parasympathetic nervous system, gastrin, we've mentioned gastrin already before, and histamine, which is a paracrine in the parietal cells. In fact, many of the antacid types of drugs that are used are in fact histamine receptor antagonists. This would be the H2 receptor that the histamine is working on. And Tagamet and some of the other drugs actually are histamine H2 receptor antagonists, and they help to stop acid secretion by targeting these histamine receptors. Pepsinogen, we've already talked about a couple of times. Its secretion is regulated in synchrony with acid secretion. So the parasympathetic nervous system, gastrin, and histamine all regulate acid secretion. They also have the similar effects on pepsinogen secretion, which it makes sense if you consider the fact that the acid is necessary for the activation of pepsinogen or conversion of pepsinogen into pepsin, that the two of them would have to be regulated in the same way and in synchrony with each other. So I mentioned the cephalic phase before. Let's take a look at a bit more detail. That the stimuli that increase acid and pepsinogen secretions, of course, we talked previously about this parasympathetic nervous system having an effect on acid secretion and pepsinogen secretion, and here's how it occurs. That the sight of foods, the taste of it, smell of it, the chewing action itself, and swallowing, all of these are considered cephalic because, of course, all of these are occurring in the head, the cephalus. So these stimuli all activate the parasympathetic nervous system, and then, of course, it in turn stimulates gastrin secretion. So let's look at that in this flow chart. So you have our cephalic stimuli here, that is the sight, smell, taste of food, etc. This is going to activate parasympathetic activity. Now, we're skipping over a lot in this era. Obviously, there's afferent traffic going from the mouth up into the brainstem, and then efferents coming from the brainstem down as parasympathetic activity. And this parasympathetic activity is going to have a couple of major effects. On the G cells within the stomach, it's going to cause gastrin secretion, which is going to increase the plasma concentration of gastrin. That will stimulate parietal and chief cells within the stomach and increase acid and pepsinogen secretion. which is going to augment the effect that the parasympathetic nervous system has itself. So that this is a redundant system and one that is mutually reinforcing. Now we have food coming into the stomach, activating the gastric phase. So the stimuli, of course, would be the presence of proteins, peptides, and amino acids in the stomach itself, as well as distension of the stomach. So we have chemical stimuli as well as mechanical stimuli in the stomach. And of course, these are working through short and long reflex pathways, and they're going to ultimately trigger gastrin, acid, and pepsinogen release. So again, we'll take a look at these steps in detail here in the flow chart. We have proteins and protein digestion products in the stomach, as well as distension of the stomach. So as we said here before, both chemical and physical stimuli. These are going to, the chemical stimuli will clearly trigger the chemoreceptors, and the mechanical stimuli will stimulate the mechanoreceptors, which will detect and have the appropriate response to their stimuli, activating both short and long reflex loops. Ultimately, both these reflex loops will again work on G cells, just as we saw in the cephalic phase, increasing gastric secretion and plasma concentration of gastrin, which again will work on parietal and chief cells, and again cause acid and pepsinogen secretion, as will the reflex pathways themselves directly activate the parietal and chief cells. So just as we saw in the cephalic phase of regulation, in the gastric phase, we do have a redundant and augmenting pathways that are involved in stimulating secretion. But we also need to inhibit secretion, because we don't want digestion to continue beyond what it needs to, especially when you consider the fact that, as I said before, one segment of the GI tract can try to inhibit the upstream segment to prevent it from shuttling more material down before the lower segment is ready to deal with it. So with that, we need inhibition of secretion. So the gastric phase, when food is exiting the stomach, it's removing stimuli for secretion of the enzymes within the stomach. So of course that would make sense, that as food is leaving the stomach, you're removing the stimuli causing the secretion of the digestive enzymes, so that will inhibit the secretion. As well as an increase in the acidity is going to inhibit gastrin release. And actually this should be rephrased, and an increase in the pH, because we're getting rid of the acid material as when the stomach is the food material is leaving the stomach. Intestinal phase for an inhibition of secretion. As the food is entering the duodenum, it's going to cause increased osmolarity within the lumen of the duodenum itself, increased concentration of fats and acid, and going to, of course, cause an increased distension of the duodenum. And this is going to work through long and short reflex pathways to inhibit acid and pepsinogen secretion. So again, stimuli within the intestine go back to inhibit the stomach, to reduce the delivery of chyme down into the small intestine, to give the small intestine more time to absorb the material that has already entered it. So let's look at the secretion of pancreatic juices. We have acinar cells as well as duct cells. So the acinar cells are those that are out here at the end of the ducts that are actually involved in synthesis and secretion. And the duct cells, of course, are involved in movement of the secretions, ultimately into the lumen of the digestive tract. So the acinar cells produce very small volumes of the primary secretion, in many cases zymogens. So these can consist of the zymogens, digestive enzymes, as I said, as well as water and electrolytes. The duct cells basically deal with large volumes. They are injecting large amounts of a bicarbonate-rich secretion that is helping to move along the secretions that were produced by the acinar cells. So we'll look at some of these secretions. There's cholecystokinin and secretin. These are primary stimulants for secretion of these enzymes. And enzyme and bicarbonate secretion is regulated somewhat independently, so that enzyme secretion, does not necessarily go hand in hand with bicarbonate. But remember, it says somewhat. It's not an exclusive. So the composition of the pancreatic juice varies based upon the luminal contents. And depending upon the chemistry of the luminal contents, there may be more enzyme or more bicarbonate, depending upon these variations. Cholecystokinin is going to stimulate acinar cells to secrete enzymes. And it is a fairly weak or small stimulant for bicarbonate release. Now, again, keep in mind that the bicarbonate is largely coming from the duct cells. So CCK potentiates the effects of secretin on bicarbonate release. Well, secretin stimulates the duct cells to secrete bicarbonate. So secretin is the primary stimulus for bicarbonate secretion, whereas CCK is less so, but CCK does potentiate the effect of secretin. And whereas CCK is a very weak stimulus for bicarbonate release, secretin is a weak stimulus for enzyme release. So the primary effect of CCK is to secrete enzymes. The primary effect of secretin is to secrete bicarbonate. And each of these two enzymes, hormones, augments the effect of the other on its secondary effect. So a main effect of CCK on enzymes is a weak effect on bicarbonate, but it can augment the effect that secretin has on bicarbonate. Secretin has a major effect on bicarbonate, a minor effect on enzyme release, but again, it can potentiate the effect of CCK on enzyme release. The stimuli for both of these, for CCK, it's fat and amino acids in the duodenum, so of course lipids and proteins entering the duodenum. For secretin, it's the acidity in the duodenum. So, bicarbonate-rich fluid secretion. We have acid entering the duodenum. This, of course, is signaling that chyme is coming in from the stomach. This is stimulating chemoreceptors, that is, receptors that are sensitive to the presence of the hydrogen ion, that is, the acid, working through both short and long reflexes, stimulating endocrine cells to increase the production of secretin, which, of course, is going to increase the plasma concentration of secretin. Therefore, there is more secretin reaching pancreatic duct cells, which is going to stimulate the secretion of bicarbonate-rich fluid into the duodenum, which is, of course, going to neutralize the acid, decreasing the acidity. And, of course, this is an example of negative feedback because it was an increase in acid in the duodenum that triggered this response. Bile, we've talked about before. It's very important for breakdown of fats, and it is also regulated. Secretin stimulates bile secretion from the liver, and CCK stimulates the gallbladder to contract, which of course is going to increase the actual amount of bile that is being released into the duodenum. And it stimulates the relaxation of the sphincter of Oddi. So by contracting the gallbladder and relaxing the sphincter, it's going to increase the amount of bile that is being delivered into the duodenum. The stimuli, we have first increased acid, which of course is telling the duodenum or the receptors there that chyme is coming in from the stomach. This is going to stimulate the release of plasma secretin. We've already talked about that before. This is going to stimulate the liver to actually secrete more bile. Also, the increased concentration of protein, digestion products, and fats. Well, of course, this, is a chemical stimulus indicating that there is processed food, chyme, coming in from the stomach. This is going to increase plasma concentration of cholecystokinin, which is going to do two things. Going to stimulate the gallbladder to contract at the same time that it's stimulating or inhibiting actually the muscle tone in the sphincter of Oddi, causing it to relax. So this, of course, is going to increase the amount of bile that's being squirted into the digestive tract. So we've increased bile secretion, we've increased its delivery into the duodenum, So the amount of bile concentration, of course, increases, which is going to neutralize the acid. Again, negative feedback here, and help to emulsify fats, which of course is going to chemically assist the digestion of the material that was coming into the duodenum. Now, there's a great deal of fluid movement we're talking about here, a lot of secretions. So the amount of fluid that, and we've talked about this before in a previous slide, previous lecture, the amount of fluid that's entering the lumen of the GI tract daily, we ingest about 2 liters. Add to that a fair amount of saliva and gastric juice, bile, pancreatic juice, intestinal secretions. The total is about 9,000 milliliters. 7,000 of it is digestive, and 2,000 of it is, of course, consumed. And this is offset by fluid leaving the lumen of the GI tract on a daily basis. We have a total of 9,000 mils coming in. 8,500 mils are being absorbed in the intestine. The colon is going to absorb another 400 mils. So we actually have, for 9,000 mils that came into the system, 8,900 mils are going to be absorbed, leaving about 100 milliliters being excreted in the feces. So the system is very efficient at reclaiming much of the water that was not only ingested, but of course far more of it was actually utilized for digestion. So let's take a look at regulation of motility. First of all, chewing of food. Chewing is both a voluntary and involuntary system in terms of the control. We can consciously choose to chew, but the autonomic nervous system, once we have food in our mouth, activates an involuntary control so that we don't actually have to concentrate on every single contraction of our jaw muscles. So it's a cyclical reflex. Normally, your jaw muscles are active to hold the mouth closed. When food enters the mouth, of course, it's going to inhibit jaw muscles to allow the mouth to be opened. The jaw dropping is going to relieve the pressure on the food, which is going to allow the jaw muscles to contract, which then when you contract the muscles, it's going to restore pressure on the food, which will then inhibit the contraction. So, of course, we end up with this very cyclical type of process going on based upon contraction and relaxation of the jaw muscles, relieving versus restoring pressure on the food. Once food is chewed, it has to be swallowed. So the chewed food and the saliva together we refer to as a bolus. The tongue moves the bolus back to the pharynx, which initiates a swallowing reflex. The integration center for this reflex is in the medulla oblongata, what's called the swallowing center of the medulla oblongata. Several steps to the swallowing reflex. It would take several slides to look at it. First of all, we have the bolus of food in the mouth. Change color here so it shows up. Bolus of food in the mouth. It descends to the pharynx, pressing on the epiglottis. So it comes down here and presses on the epiglottis. It will do several things. First of all, the epiglottis is a flap of tissue that covers the glottis so food does not enter the trachea, so it prevents you from aspiration of food. But it also inhibits inspiration. By reducing breathing while we're swallowing, it reduces the likelihood of actually inhaling some of the food. Next, the upper esophageal sphincter relaxes and the bolus enters the esophagus. So the bolus moves downstream, and then the sphincter closes behind the bolus. The bolus stretches the esophagus, therefore triggering peristalsis, which is a rhythmic movement of contractions that helps to push the bolus further down the esophagus. So the peristalsis propels the bolus to the stomach. It takes about 9 seconds. This is something you're very aware of when you drink something that's very hot, and you can pretty much feel it scalding your esophagus all the way down. This 9-second delay that is why you can be so aware of that. Once the bolus arrives at the stomach, it triggers relaxation of the lower esophageal sphincter, which is, of course, going to allow the food to enter the stomach. As the food enters the stomach, if necessary, you can have second peristaltic waves coming down the esophagus to help propel this piece, especially if you had a very large bite of food, didn't chew it enough, ultimately to complete the movement of the bolus into the stomach. Once it gets into the stomach, of course, you're going to need GI motility to process this food. So GI motility refers to the movements of the wall of the stomach itself, as well as when we talk about peristalsis, once we get down into the small intestine. And the motility is due primarily to contractions of the muscularis externa, which is the outer muscle layer of the stomach. And then the functions are twofold: to mix the food up, as well as to propel it further down the stomach toward the small intestine. In order to do that, we have electrical activity in the smooth muscle of the GI tract. These are spontaneous slow waves of depolarization, which are referred to as slow waves. And these are what are shown here. These oscillations going more depolarized, less depolarized. And they not only oscillate up and down, but they increase with time. And of course, some of this depends upon the food coming in. The frequency of the waves is referred to as the basic electrical rhythm. So how rapidly these waves occur is what we refer to as the basic electrical rhythm. And of course, that can vary in different areas of the GI tract, as well as depending upon how much food is there. The height, that is the magnitude of these waves, is affected by neural and hormonal input, which of course are themselves controlled by both chemical and mechanical stimuli. Parasympathetic nervous system, keep in mind when you talked about autonomic nervous system, we refer to the parasympathetic nervous system as the rest and digest system. So with that, it makes sense that the parasympathetic nervous system would excite this rhythm, because as we increase this rhythmic rate, we're going to increase the number of action potentials that are ultimately being generated. And of course, greater number of action potentials is going to produce greater frequency of contraction. So it makes sense that the sympathetic nervous system would excite this, and therefore that the sympathetic nervous system would actually inhibit this electrical discharge. So let's look at the relationship between the electrical discharge of the system and the mechanical events. So in the stomach, larger depolarizations produce stronger contractions, which makes sense. And then the action potentials that ride on top of this basic electrical rhythm provide even stronger action potentials. In the intestines, the action potentials are required for the force of contraction. So it's not only important what this basic electrical rhythm is, but also how many action potentials are produced in both cases to produce stronger contractions. And then the intestines, the strength of the contraction varies with the frequency of the action potentials. Of course, greater frequency will occur with not only a greater magnitude of the basic slow waves, but also the duration of those slow waves. The higher they are over threshold, will produce action potentials, but the greater amount of time that depolarization, that slow wave stays above threshold, will produce an even greater frequency of action potentials, which will produce a greater strength of contraction. Now, the mechanical events are of two types, especially once we're beyond the stomach. We talk about peristalsis and segmentation. So here we're talking about the small intestine. These are both waves of contraction in the muscularis mucosae layer in response to the basic electrical rhythm. Peristalsis are waves that propel food forward, moving it further down the small intestine, whereas segmentation is a back and forth contraction that mixes the contents. So we'll talk about each of these in a little bit more detail next. It's peristalsis. There are circular and longitudinal muscle contractions. The circular contractions, of course, are going to contract the diameter of the lumen, and the longitudinal contractions are necessary for moving a bolus of chyme forward. So in the proximal segment, that is the first segment that we come to, the circularis muscle contracts and the longitudinal muscle relaxes. This is going to allow the bolus to move downstream, and because it's going to increase the diameter of the tube. The distal segment, that is further downstream, the circular muscle is going to be relaxing and the longitudinal muscle contracting, which is going to allow the food to be pushed down through this, down through the tube. And in this case, we're increasing the diameter. The net effect of this is that the contents are propelled forward. So it's a little hard to see in a static image. Perhaps this is something we should definitely look at. The animations that are on your CD-ROM, the interactive physiology. But the idea here is that this bolus is progressively moving further down through the digestive tract because of this rhythmic contraction versus relaxation of the two types of muscle. And this is, again, peristalsis. Its function is to move material further down the digestive tract. If you notice, in that diagram, we had different colored boli of food. And what we really want to do to aid digestion fully is to mix what was consumed along with some of the secretions. So if you think about it this way, if the red ball here represents food material and the yellow one represents some of the secretions, in order to fully digest this food, you're going to have these two mixing. And segmentation, which is another type of motility in the small intestine, is a way of getting these two to intermix and basically shaking them back and forth. So if you think about my pen moving up and down here as sort of an agitation back and forth, that's what we're talking about with segmentation. So this is basically using the circular muscle layer, and it's going to alternate contractions between one segment and another. And this is going to cause this back and forth movement that is mixing the chyme. And again, it's a way of making a more thorough mixture and facilitating the complete mixture of the ingested material with the secretions. So what are the functions of gastric motility? Well, mixing chyme as well as regulating gastric emptying, because of course that's important for delivery of the chyme down to the small intestine. Mechanisms involved are peristalsis, which is coordinated by the enteric nervous system. Waves of peristalsis go from the upper body of the stomach toward the pylorus, and they strengthen. That is, these waves become stronger as they approach the pylorus. The functions in mixing the chyme is the pyloric sphincter is closed, and when the pyloric sphincter empties, the same actions function in gastric emptying. And again, because the waves get stronger as you approach the pylorus, these stronger contractions are going to facilitate the opening of the pyloric sphincter, and it's going to increase the emptying rate in proportion both to the volume of chyme in the stomach and the strengths of gastric peristalsis. But if you think about what we've already talked about in terms of regulation, the volume of material in the stomach is itself going to, through reflexes, increase the strength of the peristalsis. So those two go hand in hand. Excuse me. We also have what's called a migrating motility complex. This is a wave of intense contractions. It travels short distances, and it occurs between meals to clear your stomach. You know, when we talk about your stomach growling, this is the what it is that's actually occurring, and you can actually hear this as the muscles contracting and the walls of the stomach rubbing against each other. If you think about the fact you're trying to empty the stomach completely to clear it, you could actually hear those rugae rubbing up against each other during this migrating motility complex, causing this growling noise and sensation. Gastric motility is regulated to regulate the force of the contractions. Gastrin increases the force. CCK, secretin, and GIP all decrease the force of gastric motility. This again was summarized on the table early on in this lecture. And we've already talked about phases of gastric regulation. We'll talk a little bit more about some of the things that can activate these. The cephalic phase can be excited by anger and aggression, but it can be inhibited by pain, fear, or depression. So if you think about what the cephalic phase does, anger and aggression all increase the activity of the cephalic phase. Pain, fear, and depression all inhibit the activity of the cephalic phase. That makes sense. You know, if somebody is in pain or afraid or depressed, they generally have a lessened appetite. Well, this is largely through the inhibitory effects of these stimuli on the cephalic phase. Excitatory stimuli for the gastric phase is distension of the stomach. So as food enters the stomach, stretches the stomach, causes distension, and this, of course, activates the gastric phase, which makes sense. It's going to increase the digestion of the material as it's entering the stomach. Inhibitory stimuli for the intestinal phase are distension of the duodenum and the osmolarity and acidity and fat content within the duodenum. Vomiting is a special case of regulation of GI function, especially motility. Stimuli for it are, of course, illness, emotions. If you think about it, if somebody gets very anxious, can feel nauseous, pain, distension of the GI tract, rotation of the head, that's motion sickness, ingestion of certain substances, what we call emetics. The word emesis is a technical word for vomiting. So a substance that causes emesis would be called an emetic. And it is a reflex that's mediated through the vomiting center of the medulla. In many cases, its intent is to defend the GI tract against the ingestion of something that may be toxic. So now we'll take a look at the motility in the small intestine. The purpose of the motility in the small intestine is to mix and propel the chyme. Segmentation, as we said already, mixes the chyme, whereas peristalsis propels it. There's also a migrating motility complex within the small intestine, which occurs again between meals. It is again intense contractions that travel short distances to help sweep the intestine clean. And motility in the small intestine is also regulated. Distension of the small intestine, as you might imagine, increases motility because distension would indicate an increased delivery of chyme into the small intestine, in which case we need greater motility to help process that chyme. Autonomic nervous system, again, it makes sense. The parasympathetic nervous system, the rest and digest system excites motility because it's going to increase digestion. And if the parasympathetic nervous system excites, then it makes sense that the sympathetic nervous system would inhibit motility. Hormones that stimulate motility, gastrin. Take a look at some of the reflexes in the small intestine. There's what's called an intestinal reflex, meaning that it initiates in the intestine and its effect is also in the intestine. Injury or severe stress inhibit intestinal contractions. So this is bodily injury or just mental stress, anxiety, are going to inhibit intestinal contractions. It's also what's called an iliogastric reflex, coming from the ileus, affecting the stomach. Distension of the ileum inhibits gastric motility. So this, of course, is going to be a way of helping to provide negative feedback to slow the emptying of the stomach into the small intestine. And likewise, there is a gastroileal reflex, which works in the other direction. Presence of chyme in the stomach increases the motility in the ileum. Now, it's unusual that you would actually have chyme coming back into the stomach, but as the chyme builds up in the stomach in preparation for emptying, we need to increase the motility in the ileum downstream, of course, from the duodenum, so that the material in the duodenum can be pushed further downstream and allow the chyme in the stomach to be released into the duodenum. In the colon, we also have motility. Again, function is to mix and propel. There are two types of motility, what is called haustrations, which is what causes the mixture of the material. And it's the proximal colon because, of course, that's where we're receiving material that has not yet become fecal matter. And it's very much like segmentation, but it's much slower. And then what's called mass movement, which is to propel the bolus toward the rectum. And this is similar to peristalsis, but the contraction lasts longer than the relaxation. And this is partly because besides mixing and propelling, we also want compaction. That as we're taking moisture out of the fecal matter in the colon, we also want to compact the material into what will eventually become feces. Regulation of colon motility, there is a colonocolonic reflex, where distension of the colon in one area causes relaxation in the other. Of course, if you're trying to propel material down through the colon, this makes sense. That distension of the area upstream would cause relaxation downstream so that we can have propulsion of the material toward the rectum. And a gastrocolic reflex, where food in the stomach increases colonic motility. Again, by helping to increase motility in the colon, it's making more room, if you will, in the colon for material coming in from the small intestine, which will, of course, in turn prepare the small intestine for receiving chyme from the stomach. And lastly, of course, we have defecation. That is the process of relieving oneself of the feces. This is both under voluntary and involuntary control. If you remember, in the very first segment of this series of lectures, we talked about both having smooth muscle and skeletal muscle that are involved in the anal sphincters. Distension of the colon is going to activate, stretch receptors, which is going to cause contraction of smooth muscle in the rectum. This is going to increase pressure in the rectum. And several things can happen. If the internal anal sphincter relaxes, but the external anal sphincter contracts, this is going to continue to build up pressure. And now this whole process right here is the normal response as we grow as babies and become potty trained. We learn to control the external anal sphincter. So even though we are controlling that and increasing the amount of contraction of it, the internal anal sphincter is still going to relax because it's under involuntary control and it's responding to this increase in pressure. But because the external sphincter is contracting, this pressure is going to continue to build up. And at some point, it's going to build enough that it's going to cause us to relax both sphincters and engage in the process of defecation.